Students, we have to shift on to the next part of the lesson, chromosomal basis of inheritance, that is sex determination in plants. You have studied sex determination in the case of animals, a mechanism, different types. Likewise in plants also sex being determined by different ways. Now if you are taking the flowering plants, nearly 94% of the flowering plants are monomorphic in nature. 94% of the plants are monomorphic in nature. That means these plants produce only one type of individual. That individual produces a male sex organ named the stamens and the female sex organ called, just you see that one, pistil. Such plants are called monomorphic plants. Okay, they produce only one type of individual. That individual produces a male sex organ, the stamen, and a female sex organ, the pistil. Then what do you mean by dimorphic plants? Nearly 6% of the plants have different sexes. That means the male plant and female plants are different. So nearly 6% of what we have, the flowering plants, have two different sexes. That means we have a separate male plant and a separate female plant. Now the male plant produces only the stamens. Why? The female plants produce the pistil, the female part. Okay, this is what we have, monomorphic plants, dimorphic plants. Simply we have, so here this is bisexual and this one what we have actually different sexes, unisexual plants. Now researchers normally very much interested to observe sex determination in the case of plants. One person by name C. Allen in 1970 discovered sex determination in plants. You know, sex determination is a complex event or a complex phenomenon. So the complex phenomenon is being determined by either the genes, or we have the hormones, or we have environment. So we have chromosomal theory of sex determination having the genes, hormonal theory of sex determination, or environmentally controlled sex determination. Have you come across sex determination by environment in one plant by name horsetail? Horsetail. So it is a pteridophyte by name XII. Horsetail. This is a nickname. A pteridophyte, commonly called as actually horsetail, XII. Now, here the sex is normally controlled by the environment. So, in the case of horsetail, environmentally controlled sex determination is C. A plant which is growing in good, what is called condition, good condition. A plant which is growing in good condition develops into female. A plant which is growing in stress condition, which develops into male. So anyway, in the case of horsetail, the sex is being determined by the environment. It is one example where sex is influenced or determined by the environment. So a plant which is growing in good condition develops into female, and a plant which is growing in stress conditions develop into male. So one example, a specific example where sex is being determined by the environment. So we have to go for the sex determination in different kinds of plants, one by one, okay? Now let's see sex determination in the mechanism of sex determination in different plants. Number one, I am taking for example Xylem latifolia. That's a common name. Another name also there, Melandrium album. So in the case of this plant, we have three distinct regions in sex chromosomes. That is X, Y, X and Y. Now you see that one, the Y chromosome determines the maleness. The X chromosome is specific for femaleness. The X and Y chromosomes have different segments. They determine the sex. The different segments are 1, 2, 3, 4, 5 and so on. So that's about what we have sex determination in the case of Xylem latifolia, also called Melandrium album. Simply as in the case of human beings, X for female, Y for male, and X and Y have different segments determining the sex. Okay, the segments are 1, 2, 3, 4 and 5. Now let's see the mechanism of sex determination in papaya plant. So researchers normally recently just discovered the sex chromosomes in Hawaii. They discovered sex chromosomes in Hawaii. So the sex chromosomes were discovered by the scientists, by the researchers in Hawaii recently. In the case of papaya plant, what is called carica papaya. The diploid papaya plant has actually 36 chromosomes. So the number of chromosomes that is in diploid organism is equal to 36. Out of 36, 17 pairs are autosomes. That means 34 chromosomes are autosomes and one pair that is the sex chromosome. That is either XX or XY. So 17 pairs autosomes and one pair the sex chromosome. Now there are two types of plant. The male plants have XY chromosomes along with 34 autosomes. The female plants have actually 34 autosomes plus XX chromosomes. Now normally the sex chromosomes are similar to that of autosomes because the sex chromosomes evolve from autosomes. But the X and Y chromosomes are functionally different and functionally distinct. Though they are looking like autosomes, if you are taking the X and Y chromosome and both are functionally distinct, that means have different functions. Now the X chromosomes normally carries the genes for the development of female sex organ. Okay, so the X chromosomes are normally responsible for, or they have the genes, they carry the genes for the development of what is called the female sex organ, namely what we have the pistil. Whereas Y chromosomes bears the genes or carry the genes, they are responsible for the development of what is called the male sex organ, namely the stamens. This is one aspect of what is called sex determination in papaya. But actually, in papaya plant, sex is determined by a series of 3 alleles of a single gene. So I'll write it down in the form of what we have a tabular column and then I'll explain. Okay, so sex determination here is controlled by a series of 3 alleles of a single gene. What are the 3 alleles? We'll see one by one. In papaya plant there are 2 types of sex chromosomes, namely X and Y. X bears the genes for the development of female sex organ and Y carries the genes for the development of what we have actually the male sex organ. Now in the case of papaya plant, sex is determined by, or sex determination is controlled by a series of 3 alleles. The alleles are M, M1 and M2. M just recessive, M1, M2 are the dominant of a single gene M. So 3 alleles in different forms are responsible for controlling what we have the sex determination in papaya plant. We have different combinations and such different combinations can produce what we have either male or female, or in the case of sterile plants we can see. In some cases we can also get what we have bisexual. So different combinations of these 3 alleles produce either male plant or the female plant or the bisexual or sterile plants. Now let's see the combinations in the tabular column. I am taking what we have the genotype, either dominant or recessive, modification, that is nothing but the expression and then what is the nature of the sex produced. If both are recessive genes, that is homozygous recessive, and these 2 genes normally restrict the development of maleness, so the individual becomes female. Then if you are taking for example heterozygous condition of, that is dominant M1 and recessive M, the results normally actually the male. This is because this heterozygous condition induces maleness. The heterozygous condition induces maleness. Suppose you have another heterozygous condition with the dominant gene M2 and a recessive gene M1. That one normally induces both the sexes, both male and female organs are developed, and such a plant is called as a bisexual plant, which is normally rare in nature. Suppose you have the homozygous dominant condition or heterozygous dominant condition, we have different effects. For example, homozygous dominant M1, M1, homozygous dominant just M2, M2, and heterozygous dominant M1, M2. In all these three cases, we have the result. The plants produced are inviable, unable to produce what we have the gametes further development. So these plants are sterile in nature. So homozygous dominant M1, M1, homozygous dominant M2, M2, then homozygous, not homozygous, heterozygous dominant, then all these three conditions resulted the development of inviable plants, and these plants are unable to reproduce, hence they become sterile. So here we have the different combinations which resulted either the female plant or the male plant or the bisexual, or we have the sterile plants. Okay, that's about what we use sex determination in papaya, and I mentioned already we have three different alleles of a single gene responsible for the determination of the sex. Okay, now we have to go for the sex determination in the case of maize plant. Let us see the mechanism of sex determination in maize plant, that is A maize. Now the maize plant is monoecious. It is an example for monoecious. That means both male and female flowers are present on the same plant. Both male and female flowers are present in the same plant. That's why it's called monoecious. The maize plant has two types of inflorescence. One is found at the terminal end. That is called terminal inflorescence. Now this terminal inflorescence bears staminate flowers. That is a male flowers. Staminate flowers, nothing but you have the male flowers. Only stamens are present? No, just actually the pistil. And they develop from the shoot apical meristem, as they are found at the tip of the plant, the shoot. They develop from what is called the shoot apical meristem. Now in contrast to this one, there is another type of what we have, that is inflorescence, that is called lateral inflorescence. The lateral inflorescence bears the pistillate flowers, that is a female flowers. It bears pistillate flowers, and it is developed from axillary bud. So if you compare the differences, now the terminal inflorescence and the lateral inflorescence, it bears staminate flowers representing the male, and this one bears the pistillate flowers representing the female part, or female, actually the flower. The flower, the meaning for them is smaller ones, flower larger one. If you have in the inflorescence, the flowers are formed in what we have in the form of spike, smaller ones, then we can say that is a florence. Now that is what we have, the terminal inflorescence is developed from what's called the shoot apical meristem. And this one is developed from what's called axillary bud. Now, the terminal inflorescence is otherwise called as tassel inflorescence. Tassel. Now, the lateral inflorescence is otherwise called ears or cob. Ears or cob. Now, I'll go to the diagram now. See that one, we have, this is a maize plant. What we have, a monocot plant. Now, we have two types of inflorescence. One inflorescence is formed at the terminal, actually apex, and that is called terminal inflorescence, otherwise called as a tassel. It contains the male staminate flowers, that's why it's called as actually the male one, staminate flowers. Now another one which is developed from the what is called the axillary bud, that is called the ear or cob. It is otherwise called lateral inflorescence arising from the axillary bud of the leaf. Now if you are enlarging what we have, the maize fruit, we can say the inflorescence develops into the entire maize or corn. Now it is being covered by coverings called the husk. Okay, now at the terminal end we have fine, shiny, thread-like structures in bunch, and each fiber is connected to one what is called ovule or one ovary. So these fibers represent what we call the styles, because we have many ovaries, many flowers or many florets. Each one is having an ovary, and each ovary, you know, that one is connected to the style. The styles are long, fine, thread-like, shiny structures. Hence the name silk. The word silk refers to the long, thread-like style leading towards the ovary. Now, after fertilization, you know that one we have formed the seed. In the case of maize plant, the seed is nothing but the fruit only, what we have the caryopsis. So I represented one what is called actually inflorescence with many seeds or caryopsis fruit. Now this is nothing but the ear or cob, the one which is enlarged here. This is after fertilization, after the formation of the what we call the seed, or we can say the caryopsis fruit. Okay, this is about the maize plant. What are the types of inflorescence present? Now normally I mentioned about, actually we have the male and female flowers are present on the same plant, hence called what is known as monoecious. Now the unisexuality of the flowers, we have either staminate flowers or we have pistillate flowers. The unisexuality of the flowers, because of the unisexuality in the case of maize occurs because we have either male flower or the female flower, that is called unisexuality. So the unisexuality in maize occurs due to selective abortion. That is what we call the abortion means, you know, that one destruction, as in the case of abortion of the child. Likewise, because of the selective abortion of either the stamens in ear flowers, because the ear flowers contain only the female part. You know that one, the female flowers, otherwise called the pistillate flowers. You see that one? So selective abortion of what's called stamens in ear flowers. That's why they have only the pistillate flowers and no stamens form. Or because of what's called the abortion of what is called pistil in tassel flowers, because the tassel flowers contain only the male organ, the stamens, so the pistil is being aborted here, and in the case of ear flowers, the stamens are aborted. So that's why the maize plant is unisexual. That's why I mentioned here, maybe I asked a question also. What is the reason for unisexuality in maize? It occurs due to selective abortion, selective destruction of what? Number one, stamens in ear flowers. That's why it contains only the female flowers, or we have the pistillate flowers, or we have the abortion of pistil in tassel flowers. That is nothing but the male inflorescence, where we have the staminate flowers only present. This is because of the abortion of the pistil or the female part. Okay, this is one thing. Now we have to go for how far the difference between what we have the monoecious and dioecious plants are caused, or we can say the monocarpic or dicarpic or anything else. So what is the reason for the development or for the differences between what we have actually just these two types of the plants? That is what we call the unisexual plants. Let's see now. Which makes the difference between the monoecious and dioecious plants in maize. So mostly the maize is monoecious. We have actually the flowers of male and the flowers of female that is present on the same plant. But a rare instance we also have a dioecious condition. Which one makes normally the difference between monoecious and dioecious condition? This is because of a substitution of two single gene pairs. The difference between monoecious and dioecious plant is made because of a substitution of two single gene pairs. That is BA, another one what we have TS, both are recessive. This BA for a barren plant. The barren plant is nothing but the female plant, the female part. And we have TS for tassel seed. That is nothing but the male part. We have seen already tassel inflorescence. So we have two single gene pairs. These are the two single gene pairs. They are responsible for making the differences between the monoecious and dioecious plant. The dioecious plant is now rare in maize. So these genes are responsible for making the differences. The another statement, suppose the allele, namely BA, for barren plant when in homozygous condition makes the stalk staminate, produces the staminate by eliminating the silk and ears. I mentioned already the silk is nothing but the style, the ears are nothing but actually you have the flowers, pistillate flowers. So when the allele BA for barren plant present in homozygous condition and makes stalk staminate, production of staminate by eliminating silk and ears. That's why we have the tassel plant formation. Similarly, another allele. Another allele for tassel seed, we have seen already TS for the development of what we have the tassel seed. Transforms tassel into pistillate structure. This is the gene responsible for transforms what we have the tassel into pistillate structures. That's why which produce no pollen grains. So the development of what we have the staminate flower and then pistillate flower is because of selective abortion. This is because of the presence of the two. Just what we have the single gene pairs BA and then TS. In different combinations, they produce either the male or the female or reduced female, etc. Let's see actually the sex expression. The result of sex expression because of the combination of these alleles in the form of tabular column. Normally, most of these mutant forms, most of the mutant forms normally cause defects in the synthesis of gibberellin hormone. The gibberellin hormone is responsible for the suppression of stamen formation, suppression of what is called stamen growth in the case of ear flowers. In the case of ear flowers means the female part. So it is a function of the gibberellin hormone that is being expressed by what we have these genes, and these suppress the gibberellin hormone, suppress not only the staminate formation or staminate growth in the case of ear flowers. So the genes are expressing either the gibberellin hormone or not. Accordingly, we have developed what we have the male or female flowers. That is, we have the inflorescence, like terminal inflorescence or tassel, and we have the lateral inflorescence or what we have the comb or we have ears. Now I will give the tabular column to show the result of what we have the sex expression by the combination of different alleles. Now the following table shows the resultant sex expression based on the combination of alleles. So different combinations of the two alleles BA and TS produce either in dominant form or in recessive form different sexes, either male or female, or we have what is called rudimentary female or monoecious condition. Now take the first one. We have double recessive. Both BA and TS are found in recessive form, double recessive. And here what is happening, the modification, the expression, lacks of silk. There is no style present. No style present, lacks of silk on the stalk, but transformed tassel into pistil. The tassel being transformed into pistil, but not fully because of the absence of style or the silk. Now we have the rudimentary female. The female is not fully developed because of the absence of silk or what we can have, that is the style. Now the next one, see that one is actually a recessive, another one a dominant. Now the barren gene, just actually gene for the allele for BA is normally recessive, and here I represent the superscript plus representing what is the dominant. So the BA is recessive and we have the TS is dominant. Again we have lax silk, absence of style, and only we have the tassel. This is because of the presence of the dominant gene. So it is male because the tassel is always male, possessing what is called the male flowers. Now we have, now this one BA is in dominant form and TS is in recessive form. So the first one is dominant and the second one is recessive. And this is because of that one, it bears comb and lacks tassel because the BA is responsible for the development of the feminine character. So it bears comb, that is inflorescence, ear inflorescence, and lacks tassel. So this is a normal female because of the presence of what we call the dominant gene BA. Now here both the genes are in dominant form. That's called double dominant. So because of the presence of dominance of both the genes BA, that is alleles BA and then TS, we have the formation of both, that is goes for tassel and cow, and such one is called monoecious. So monoecious means not actually that one monoecious condition, a rare condition. And we have the formation. So bisexual flowers, we can say bisexual flowers. The plant is monoecious means the presence of female and male flowers on the same plant. If actually the flower is bisexual, we can also say that for monoecious, the presence of both male and female sex organs in the same flower. We have different meaning. Anyway, just about the expression of the different genes in different what we have format results in the formation of different sexes, either rudimentary female or male or normal female or monoecious. That's about what we have the sex determination in the case of maize or corn. See, the superscript plus always referring to what we have the dominant character. Even it is given recessive form the letters, but if superscript plus is given, it always refers to the dominant form. Okay. So with that I concluded about this one, sex determination in the case of maize, and one more paragraph regarding the sex determination in the case of one bryophyte, Spirocarpus donally, and that one I will conclude quickly. Another plant in which sex determination was proposed or actually discovered, a bryophyte called Spirocarpus donelli. Spirocarpus donelli, a bryophyte, the common name for that one, bottle liverbot, spreading on the surface of the substratum, a prostrate growth. So this plant is a bryophyte. Sex determination was first described in that plant only. Now normally the thallus is a gametophyte, which is heteromorphic in nature. So if you are taking the chromosome also, the chromosomes are heteromorphic. Two different types of chromosomes, X and Y. That's the meaning for heteromorphic chromosome. So sex determination was first described in what we have in that plant. Now it has what is called heteromorphic chromosomes, X and Y chromosomes. Now the gametophyte, which is haploid, and also heteromorphic, because we have female gametophyte and male gametophyte. They are different in their external morphology. That's why I mentioned gametophyte, haploid. Heteromorphic means we have two different types of gametophyte. One, the male gametophyte with Y chromosomes, another one female gametophyte with the X chromosome. That's the meaning for just actually, you see that one, heteromorphic chromosomes, different chromosomes, X and Y. Heteromorphic, what we have actually gametophyte, male gametophyte with what we have Y chromosomes, and female gametophyte with X chromosome. Now, the male and female gametophytes I mentioned already, haploid nature, having a number of chromosomes, namely 8. So n is equal to 8. They have just actually haploid. Normally, usually in the case of gametophyte, we have n number. In the case of sporophyte, we have 2n number. So here the male and female gametophytes are haploid with 8 chromosomes. So n is equal to 8. The 7 chromosomes are called autosomes. The 7 chromosomes are called autosomes. The 8th chromosome in female is called X chromosome. The 8th chromosome in female gametophyte is called X chromosome, which is normally larger than the 7 autosomes. Now in the case of male, we have 7 autosomes and we have 1 Y chromosome. The male gametophyte has 7 autosomes and 1 Y chromosome. Now the Y chromosome is normally considerably smaller than the autosomes. The X chromosomes are larger than the autosomes. The Y chromosomes are smaller than what we have actually the autosomes. Now, if you are taking actually a sporophyte, we have alternation of generation. The gametophyte is haploid and another one what we have sporophyte, which is diploid. The sporophyte is always heterogametic because it produces two different types of gametes. The sporophyte is always heterogametic. The meaning for that one, it produces two different types of gametes. Suppose we are taking what's called a sporophyte with XY combination. XY combination. That XY combination sporophyte produces two different types of meiospores. Meiospores means the spores which are produced by means of what is called actually reduction division. Now the sporophyte is diploid in nature. A sporophyte with XY combination produces two types of meiospores. The spores which are produced because of meiosis are called meiospores. Now some meiospores with the X chromosomes and some meiospores with Y chromosomes. Some meiospores with the X chromosome, some meiospores with Y chromosome. The meiospore with the X chromosome develops into female gametophyte which is haploid. And similarly, the meiospore with Y chromosome develops into male gametophyte with n number of chromosomes. So in this way, the sex is determined in the case of what we have the liverboard, normally called as a water liverboard, Spirocarpus donelli. It is the first plant in which the sex determination was described. So with that I concluded about what we have the sex determination in different plants, as in the case of papaya, or in the case of maize, or in the case of this plant, just what we have the liverboard, water liverboard. And I conclude about that one. We have to go for the next one regarding the mutation in the next class. Okay, thank you.